extraction laboratory. It was published earlier this year in Genetics. RNA additive is a post-transcriptional process reported RNA nucleotides. Two types of editing have been reported. They are A2A and C2U change. A and U can be converted to G and T in the CDN. So in the CDN, the A2A and C2U change become A2G and C2T. And A2A editing have been reported that's catalyzed by ADA family. So all the members of ADA family act as the arm of the secondary structure, where the A's can be edited. And for the C2U editing, it's catalyzed by AppleBack1. So there is a motif called a Morin sequence that can localize AppleBack1 and ACNF to a position where the C can be edited. But so far, whether other factors involved in this process is not clear. So the goal of my project is to identify new factors that control editing efficiency and determine which transcript can be edited and which A's can be edited. Actually, except the A2A and the C2U editing and other types of editing have been reported. Here I call them non-canonical editing sites. Before I do the genetics mapping, I work on developing a pipeline to identify new editing sites. And I found most of those sites are the artifacts from the analysis, such as for most of the sites reported in this paper, most are from the improper alignment at the splicing junction regions. And except that, other factors can produce artifacts, such as if the sequence not poorly random, they can produce strand biased size. And if the alignment is not very improper at the parallel genes, they can also produce artifacts. Other factors including the antisense transcripts, incomplete annotation of the reference genome, somatic mutations, random sequencing errors. So I developed a pipeline to solve those problems. It was my first work, published in 2012, and after it's published, I improved multiple times for different projects. It's also the basis for the genetics study. So here I'll give you a quick review of the pipeline. So there are several advantages. First, it can split long reads to short fragments, so it can tolerate more mismatches since editing site towards to cluster together. And also it can align to splicing junction database and reference genome in parallel. So it's very efficient alignment. And also solve the problem at the splicing junction regions. And the splicing junction database contains all combinations of each two known exons. So it can supply a relative complete reference for the reader to align to. So you will carefully do the alignment. After the initial cut, there are still many errors, and I showed in the previous slides. So I developed multiple filters to remove those artifacts that arrived from different sources, such as use Fisher exact test to remove the strand biased size. And also many other filters, such as based on the quality score, the coverage, editing ratio, remove known SNPs. Here the sequencing variation means if the sites consistently edited across different samples, so there's no variation across samples, or if the sites consistently appear as specific region of the reads, or the canonical sites cluster with non-canonical editing sites, they are all positional artifacts. So after remove those, get a small set of confident editing sites, I normally do another round of alignment using a different algorithm to ensure those regions are properly aligned. So use this pipeline, almost all the editing sites discovered are real editing sites. And since I know majority of the non-canonical sites are properly artifacts, For the genetic study, I focused on A2A and C2U editing. So the method used is QTL mapping. So here, this figure shows how QTL mapping works. So normally get the phenotype from each animal and then scan the genome of the animal using a statistical model to find the region that significantly correlated with the variation of the phenotype. So if if this region significantly correlated with the variation of the phenotype, probably this region is a causal for the phenotype. And here shows the data set I used for the QTL mapping. They are from diversity outbred mouse liver. Here are called the dual mouse. And we also got a large set of data from the founder strains. And here shows how the dual mouse derived. The purpose of developing the dual mouse is to supply a mouse population that's similar to human, heterogeneous and enriched polymorphisms. So we carefully selected 8 strains and including 3 wild-derived mouse strains to enrich the polymorphisms. So first, in-breeding those mouse, and then out-breeding. In the end, we got the dual mouse, and their genome are mixed of the 8 founder strains. So there are 2 advantages to use a dual mouse. One is enrich the polymorphisms. So when do QTL mapping, it's can narrow down to a small region. And also, the 8 founder strains were sequenced by the Sanger Institute. So it makes it easier to interpret the genome of the dual mouse. And since I know it's not very accurate to identify novel editing sites from heterogeneous samples, especially enriched polymorphisms. And also the goal of this study is to identify novel factors. So I designed this strategy to do QTL mapping on editing. Only identify novel sites from the founder samples and collect known sites from the literature and quantify each editing site using the dual mouse. And the pipeline for identifying novel editing sites is similar to the pipeline I showed in the background part. But improved by aligned to the 8 corresponding founders transcriptomes rather than aligned to one reference. And for the haplotype, we used the HMM algorithm. So we not only genotype each dual mouse, but also the founder and F1 animals. So using the founder and the F1 animals, we built the emission probability matrix for each state and used the consistent transition probability matrix and did a forward and backward HMM. And in the end, we get a matrix contain the 8 probability contribution from the 8 allele for each SNP from each sample. And here, showing an example how the data look like. So in the table are the 8 allele probability contributed by the 8 alleles for one SNP from 10 samples. So from this allele probabilities, it indicates that the 10th animal is a homozygous animal, and the others are homozygous animal, heterozygous animal, contributed by different 2 alleles. And this is the basis for the building the kinship additive model for mapping and also the basis for interpreting the result. And now here is the additive model. The E here is the number of edited reads. And this part is the contribution from the 8 alleles. And the gamma here is the kinship that's calculated based on the 8 allele probability from 2 animals. So it represents the relationship between 2 animals. And we also put the total number of reads at each site as a covariable to remove the potential effect from the sequencing depth at each site. So using this whole pipeline, we got a very nice result. Here shows for the C2 editing site. most of those sites map to one region on chromosome 6. So here shows where the QTL is localized, and here shows where the editing site is localized. So it doesn't matter where the editing site is localized, they are all mapped to the same region on chromosome 6. And very interesting that the pattern of the 8 allele contribution is similar for all the QTLs. It's a surprising result. And here shows an example for the most well-studied C2U editing site in ApoB. It's on chromosome 12, mapped to chromosome 6. And here shows the enlarged region of chromosome 6. And this is this peak. And here shows the coefficients from the additive model for the 8 alleles. And they form 4 groups. I hope you can see clearly. So this 2, 1 group, and this 2, 1 group, and here are 3, and here is 1. And in the peak center, the only known C2U editing enzyme, ApoBac1, is there. So we think ApoBac1 is the gene that contributes for this QTL. The reason we think it's ApoBac1 because in that region, it is the only gene that's associated with C2U editing. And also most of those other genes have no expression in the dual-mass liver. And more importantly, I found the polymorphism that can explain these 4 patterns. And here shows the polymorphism I identified that can explain these 4 patterns here. I just replot the coefficients that showed in the previous figure by the bar plot. So here is the first SNP shared by CAST and PWK. It changes the amino acid. So probably this SNP increased the catalytic activity of ApoBac1, and also here marks a large insertion shared by BLAC6 and NZO. So it seems this insertion produces a non-functional ApoBac1. And also here shows one SNP shared by these 3 strains. And this SNP produces 2 different known isoforms of ApoBac1. And probably these 2 known isoforms have different activity. So here I identified the 3 novel polymorphisms that probably impact the function of ApoBac1 and then further control all the C2U editing sites. And for the A2 editing site, we got completely different phenomena. So most of the QTLs are local QTLs, and here shows where the QTL is localized, and here shows the editing site localized, and they are all local QTLs. And the interesting is that the edited gene itself in the peak region. And here show you an example. The multiple sites are in like B2 gene, and they all map to chromosome 1. And here is shows the enlarged region of chromosome 1. So this should be chromosome 1. And here are the 8 allele contributions. So PWK is the only allele that with lowest editing efficiency. And in the peak region, this gene, the edited gene itself in the peak region. And initially, we have difficulty to explain this phenomena, but as I showed in the background part, A2 editing is catalyzed by ADA family, and ADA acts as the arm of the secondary structure. So we tested the length of the arm and found that PWK strain with a shorter arm. So it seems the longer the arm, the more efficient on A2 editing. And sometimes we can predict the exact polymorphism that affect A2 editing sites. Here show you an example that one editing site in CDS2 is mapped to chromosome 2, and CAST and PWK are the 2 alleles with lowest editing efficiency. And here shows the local secondary structure around these editing sites. And next to the editing site, there's a motif shared by CAST and PWK on the secondary structure. So in the linear sequence, they are far away, but on the secondary structure, they are next to each other. So probably this SNP contributed on the efficiency of this editing site. So just a brief summary. So I developed the pipeline that can accurately predict and quantify the editing size and does QTL mapping at a single base resolution. And distinct genetic pattern were found for the 2 types of editing. And C2U editing are under control of AppleBack1, and 3 novel polymorphisms were identified. In AppleBack1, probably impacts the function of AppleBack1 and further control all the C2U editing size. And the secondary stats are very important for A2A editing size, and the data set deposited to DO and the program online with the paper. And I thank my previous mentors, Dr. Brown, Churchill, and Hives, and also the lab members and the collaborators from both the Jackson Laboratory and the University of Wisconsin, and especially then, after I left the Jackson Lab, made a lot of effort on this work. And I thank my current institute for their support to let me to come here, and thank you for your attention.